Well, this is Michael Comet at South Lewis High School once again. Several teachers across the state are still remote, which means they don't have their students in the classroom. I have the pleasure of having my students every other day. I'm very fortunate in that aspect. As a result, these 4 mandated state labs tend to become very difficult. So today, this video is going to address the relationships in Biodiversity Lab. There's a better picture of me right there. You don't have to look at me this way. This lab is lengthy, but it is probably the best of the state labs. Some would argue with me on that one. Some people swear by it, some people swear at it. Ultimately, in this lab, you're going to do several tests that are designed, I guess you could say, to try to identify a replacement for a plant called Botanicurus. And Botanicurus is fictional, it's not real. The Botanicurus, however, does have a chemical compound that can be derived from it that may cure some forms of cancer. But it is in very limited supply and grows very slowly, so we have to find a comparable equivalent. The equivalent is out of 3 other species X, Y, and Z, very interestingly named, I guess you could say. And ultimately, at the end of this lab, you're going to end up filling out a table, once again, going to hide my face, that looks like this. I gotta look around the side here a little bit. Okay, you can see over here on the side, X, Y, Z, and Botanicurus, and this is all the different tests you're going to do. So you're going to fill in this entire table. My hope is that by the end of this video, you'll be able to see every test and then determine which one is most closely related to Botanicurus. So let's have some fun now. As we begin the Relationships in Biodiversity Lab, here are the plants you're going to be working with. Botanicurus X, species Y, and way over here, species Z. Now, these are once again fictional. Well, they're real plants, but their species and in genus is entirely fictional. Botanicurus. This is the plant we're trying to replace, and we're worried about its biodiversity because it grows very slowly. So for test 1, the structural characteristics, I would say this is a very broad leaf with sharp points on the very end. Okay, if we look in, the stems themselves are somewhat, I guess you could call them hairy. Okay, so those are the characteristics you'd write down for Botanicurus. Let's look at species X. Species X has a wide, broad, flat leaf with soft, rounded edges, and they seem to be mostly bordered with a white, almost a dusting around the edge. If we look at the stem, just to be fair, it's not quite as fuzzy or furry as Botanicurus. So let's say that is species X. Species Y has that broad leaf once again, somewhat soft, rounded borders, but look at each of the leaves. Each leaf seems to have a red or a purplish ring around the center of it. To be fair, once again, let's look at the stem. Sorry about the box in the background there. A little bit fuzzy, but not as much as species, or as Botanicurus, I'm sorry. And then species Z. Species Z that seems to be kind of all over the place. It has a very odd shaped leaf, multi-lobes. Okay, multiple lobes. A little bit fuzzy looking, pointed but not very sharp, kind of rounded almost. And once again, let's look at the stem. It is kind of fuzzy again. So on your table, at the very end, you're going to record all the features you see of these plants. So once again, let me take a look at them here. Botanicurus. X, Y, and Z. Okay, we have done the structure of the plants. We looked at all these plants. Now we're going to come back and we have to look at two more features. We have to look at the seeds, and then we have to look at the stem structure of each. So let's do the seeds for Botanicurus. Kind of kidney shaped. Imagine that. They have a white spot, I guess you could say, at the dent in them. They kind of look like they're a light tan, dark tan camouflaged seed. So you can write that in that spot on your data table. The stem structure, if you were looking at it under the microscope, you'll notice a couple of things. Okay, number one, lots of cells. But look at this ring all the way around the outside. Okay, it's kind of circular in nature. Sorry about the shaking once again. Over to species X. Species X seeds are flat, somewhat light green and tan, maybe a little bit of gold mixed in. You'll notice that they are flat disc-shaped seeds. And so we look at the stem structure here, and it looks like someone just let a pencil go crazy and put little spots all around. It's very scattered throughout the stem. The vascular bundles are scattered. Species Y. Seeds are light green, somewhat flat. Some are a bit tan or gold. Some are darker than others. The stem structure. Once again, vascular bundles are very scattered throughout. There is no defined circles of these masses of bundles. And lastly, the species Z. Seeds are a dark brown with some darker brown patches. They do have that white spot right at the dent of the seed. And if we look at the vascular bundles here, although the picture got chopped off, you'll notice once again, okay, kind of a circular bundle. Okay. So to recap, Botana, light brown seeds, dark brown markings, vascular bundles in more or less a circular fashion. Species X, flat, brown, tan, somewhat gold seeds, scattered throughout. Species Y, flattened seeds, different colorations. Vascular bundles are scattered throughout. In species Z, darker brown seeds, some camouflage appearance to them almost, and the vascular bundles are in a circular pattern. This covers both the seeds and the microscopic stem structure. One of the next tests is to use this paper, which is called chromatography paper. And this is the paper chromatography test. What you have to do is, using a straight edge, I'm just using an index card here, draw a nice faint pencil line. And by faint, I mean hardly even touching the paper, just drag the pencil. You don't want the pencil really interfering much. That's all you want, really. Up near the top, we're going to put Botanicurus X, Y, and Z. Okay. Using toothpicks, and this is where it gets a little bit time intensive or time consuming. Just put a dot right on the paper of Botana. You can probably hardly even see that one. I'm just dipping it into that same extract that we used for the other test. X, kind of line it up here. Y, again, just a faint dot. And I got to get one more toothpick here. Extract Z. So I'm going to zoom in. I want you to take a look at this piece of paper. Okay, very, very faint dots. Now I'm going to hopefully get it to stay there, and we're going to do more. You can start to see these are the teacher tattoos I was talking about. You get that all over your hands. We have to do that dot of extract about 5 times. So back to the X and the Y and the Z. And we're going to do that repetitively 5 times. 1, 2, 3, 4, 5. Give it a few seconds to dry, and then we'll continue the video. All right, now we're back for the second portion of the paper chromatography test. You can see I have a little petri plate full of water, and here are those pigments, Botanicurus X, Y, and Z, that I have spotted on the line. Now that line is very faint pencil. It's nothing that's going to interfere because the pencil will not bleed. What we're going to do is I'm going to fold it in kind of an accordion style off camera. And then we'll put it back into that water, not back into the water, into the water for about 5 minutes. Okay, and the reason I have to go accordion style is so it stands up in here. This is a special kind of paper known as chromatography paper. It's very, very absorbent. Someone just dropped something heavy in the hallway, it sounds. And what you're going to see over the course of a few minutes, like I said, probably about 5 minutes. So you've got the accordion. I'm going to set it right in the water, and I'm going to take it off I'm going to shake a little bit here, and we don't need Siri, honestly. You can already start to see what's happening. Okay, the pigments are starting to creep up the paper. Okay, oops, sorry about the finger there. Okay, they're going to start creeping up the paper very, very slowly. Now when it gets about halfway up the paper, we're going to take it out. We're going to take a look and see which colors we see and record that on your lab. Okay, so now I've let the water wick up through capillary action up this chromatography paper, and the 4 pigments are still represented. What you have to do now is, starting at this point where you put the dots, look for colors. You may have to look pretty closely. You'll notice that this one has a little bit of a pink, and that kind of blends into a very faint yellow and then bluish green. X is basically pink right to blue. Y has no red, a little bit of green, and a little bit of blue. And then when you come over to Z, we have pink, kind of a yellowish transition area, and green and blue. So in this case, Botana and Z are most alike. So you're going to write that down on your data table as well. That concludes the paper chromatography test. And yes, teachers that have been around a while know that this is called the teacher tattoo. It used to be over here on the side of your hand when you were using overhead markers, and I really just aged myself by saying the word overhead. Now we're going to begin station number 5, or test number 5, test for enzyme M. Each of these little vials has the extract in it. Botanicurus X, Y, and Z. And this one takes a little bit. It's more setup and actually not getting your hands all blue and green, affectionately known as teacher tattoos. So I'm going to put a couple of drops of each extract. That's more than a couple of drops. Using a different dropper for each one, of course, because we don't want to get them all mixed up. There's our Botana. Here comes our X as soon as I can get it out here. About as much X. Y. And off camera, all I'm really doing is drawing this up in the droppers, so you really don't need to worry about that. And lastly, Z. Okay, now they look a little bit different as you go through and do this, but you'll see a definite difference in a few moments here. Okay, so there's our 4 pigments. And what we're going to do next is put an indicator powder, which is truly just baking soda, in each one, and we're looking for a chemical reaction. So here is Botanicurus. Get a definite fizzing in Botanicurus. In X, no real fizz going on there. Y, a little bit of a fizz. And in Z, very violent fizz once again. Okay, so we have a fizzing, no fizz, no fizz, and a fizz. And that is for the test for enzyme M. All right, this is going to be test 7, which is translating the DNA code to make a protein. Now you'll see we have Botanicurus X, Y, and Z listed here, and you see a small section of the DNA, yes, that's DNA, here. Now, quick review. Here's your DNA molecule, kind of like a little accordion or a twisted ladder, I tell my students, okay? When we have to create proteins, or when any organism has to create proteins, of course, it uncoils the DNA and kind of tears it in half. This is an old model, so it comes apart really easily. And what we're left with is RNA. RNA, of course, is single-stranded. This one has a bond on there to get that bond off. Okay. Then we go through 2 processes, transcription and translation. And those transcription and translations are going to give us a sequence of amino acids. But first we have to get the RNA that it produces. So CAC will become, and this is going to be very difficult, I'm reaching around my tripod and I have horrible handwriting anyway. CAC becomes G, U, G. GTG becomes C, A, C. GAC becomes C, U, G. TGA would become A, C, U. GGA becomes C, C, U. CTC becomes G, A, G. Told you I have horrible handwriting. And then CTC becomes G, A, G. Notice these 2 are the same, so the actual mRNA that's produced is the same. Now, that's pretty easy. I think you can all get that. You've probably all done a protein synthesis lab at some point during this year. Okay. Now comes the more difficult part, and that is figuring out what the amino acids are, excuse me. So, this is one of my favorite things ever. Okay, this is the universal genetic code chart. Now, you'll notice there are 2 on the screen. Okay. This is my preferred one, the circle. Probably because I played a lot of darts in college and you know, that sort of thing. This one is kind of reverse softball or baseball. I was never super athletic, okay? So I find it backwards. I find this more clunky to use than the circle, but I'll show you on both. All right, flipping back and forth, sorry. We first have to find GUG. So back to our charts. Most often on the regions, you're going to find the square chart. Okay, so first letter was G, second letter was U, third letter was G. So we kind of do the intersection thing, and GUG gives us val. Okay, so what you're going to do, back to the other paper, is put Val. Val stands for an amino acid known as valine. Okay, you don't really need to know that for this, but it's something that, you know, it is good to know. Next one is CAC. So let's go back to the chart. Okay, CAC, C, A, whoops, A, C, bring it together, you'll find that it is HIS, H-I-S. So we'll write that in. But I'm going to show you one other thing, okay? Once again, that first one was G-U-G, and let's go to my preferred one. Start at the middle, start at the bullseye. G, U, G gives us valine. Same results. Next one was CAC, C, A, C gives us HIS. Next one is GAC, which becomes CUG. So CUG becomes CUG LEU, which is leucine, by the way. ACU, ACU, THR, 3 and 8, okay. CCU, CCU gives us pro. Look, right over here, CCU gives us pro. So these two tables are identical. It's important to realize. You could, boy, that's handwriting bad, sorry. You could use either one. You could see either one on the reagents. That's fine. GAG is our next one. GAG gives us glue. Same thing over here. Not glue as in what you use in art class or something like that. Okay, I think it's glutamic acid or glutamine. GAG, GAG gives us glue. Same as the previous one. Now, you're going, I'll get rid of this once again, universal genetic code chart. And yes, there's an app for that. Now, you can go through the rest of these. I have utmost confidence that you can figure this out. And this is test 7, translating DNA to make a protein. Although it's fun, we are at the final test. This is the gel electrophoresis DNA banding pattern test. Each of these different extracts, Botana X, Y, and Z, are represented by strips of their DNA here. We have to cut the DNA, and that's exactly what happens in electrophoresis. We're going to cut it with an enzyme wherever we see 2 C's and 2 G's. It's very difficult to see, I know, but I really don't want to zoom in. I want you to be able to see the entire process. So just with a pencil, go through wherever you see 2 C's and 2 G's together, put a little mark. Following along, there's 2 C's and 2 G's, we're going to put another one. Follow along a little more, 2 C's and 2 G's. Just kind of cut lines for ourselves. Okay, so that's it. So, excuse the shaking, but here we go. Okay, we're going to clip off each of those sections with a pair of scissors. No, they're not scissors. They're a restriction enzyme that is meant for cutting DNA. And this would be added to a sample and just set them. These are the Botanicurus letters, so we're going to set them in the Botanicurus column of this table, or this gel, I should say. And then since we know it's Botana, we're not going to drop it. We can just throw the Botana part away. Okay, now we have all our pieces. And now we're going to run the gel, so to speak. And we do that by taking some tape and gently touching our sample. And we count the number of bases. 1, 2, 3, 4, 5. So for Botanicurus at 5, that's where we're going to put that DNA bit. Sorry about the wiggle there. Just like that. I've done this lab enough to know that the next section has 9. Okay, and if you want to sit there and count them out. You're more than welcome to. 1, 2, 3, 4, 5, 6, 7, 8, 9. So that will go right here. And then the last two are a bit longer, 11 and 12, I believe. Okay. Again, if you want to count it out, don't believe the old man that's been doing it for a long time. 11. And a little bit longer with some tape. And 12. Okay. So that is your first gel lane. Okay. You do the same for X and for Y and for Z. Now, I happen to know that these are all in black and white because I got a new printer. I got a new laser printer. Normally I used to print these in color and it was a lot more colorful. So what you're going to do is you're going to continue the same thing. Remember, wherever there's 2 C's and 2 G's, you cut it right here. Okay? And you cut it once again with your restriction enzyme. Tape them in the appropriate columns. And what you're going to, or excuse me, I'll call them lanes. What you're going to see is something is going to match most closely with Botana. Okay. That is it for the gel electrophoresis DNA banding pattern test. This is your final table. It's odd that it's the final table and it says table 1. However, there are 2 different types of evidence once again. Structural evidence, things you can see with your eyes, the structural characteristics of the plants, the seeds, and the microscopic stem structure. Then there's molecular evidence, the stuff that isn't quite as apparent when you look at it, doing the paper chromatography, the test for Enzyme M, the differences in amino acid sequences, and the gel electrophoresis banding. I failed to mention for the differences in amino acid sequences, you look at Botana and see which ones it has the most in common with, and you should see that it has no differences from one of the others. Once you fill out this table, you can go back and complete the questions. So that's it for the Relationships in Biodiversity Lab. We covered all 7 tests. You've done gel electrophoresis, you've done chromatography, you've looked at the stems and seeds and cross sections of plants. You've done quite a bit. Now you have to compile all that data onto that one data sheet, table 1, should be probably the last page of your lab, and see which one Botanicurus is most similar to. And hopefully we can use Botanicurus to cure cancer or whatever the fictitious scenario is. Hope this video helps you out. Have a great day.